But yeah, so today I'll be talking about inductively coupled plasma mass spectrometry, ICP-MS. I'm a research scientist here at Colorado State University. My position split between ARC and the Department of Horticulture and Landscape Architecture, just for a little bit of background. So let's dive right into what is actually ICP-MS. So ICP-MS is basically the analysis of the elemental composition of a sample. So a lot of times when people ask about elemental composition, they think of carbon, nitrogen, oxygen, and hydrogen. And those are one of the few elements that are actually not analyzed by ICP-MS, but anything else, for the most part, can be analyzed by ICP-MS as long as they're stable isotopes. And so you have different types of elements. You have something like sodium with this monoisotopic, which means it only has one isotope that you can measure, whereas you could have something like zinc where it is comprised of multiple different isotopes, such as zinc 64, zinc 66, 67, and 68, and 70, and they have all different relative intensities or abundances that are naturally occurring here on Earth. And so with ICP-MS, there's a wide range of fields that can be used to analyze that can use this technique to analyze samples. They're used in food testing, routinely in geochemistry and geology, in chemistry, in archaeology, in environmental sciences, to analyze energy and fuels, as well as clinical research and water quality and stuff like that. So these are really used as a broad range. This technique can be used in a broad range of different fields throughout. So the ICP-MS that we have here at Colorado State University is the PerkinElmer Nexium 350D, and it's located in the microbiology room C110, just for. And so this is, when you're measuring ICP-MS, this is a relative measurement method where we need to essentially calibrate the system prior to analyzing unknowns in order to determine concentration of the element in the sample. And so let's just peek in underneath the hood, so to speak, to kind of determine and to see what are all the components that are in the instrument. So here we have various different components. We start the torch. So this is where the plasma, we have a triple interface cone, the quadruple ion deflector, the universal cell, the main filtering quadruple, and a detector. And I could talk about what those things do a little bit more briefly. So in the plasma, in the torch, that's where the plasma is made, and it atomizes and ionizes ions. The triple cone interface defines a narrow ion beam. This quadruple ion deflector, or QUID, is able to turn all the ions, positive ions that are created, into 90 degrees. And so any photons or neutrons that aren't ionized will go towards the back of the instrument. So this helps with background and sensitivity. We also have a universal cell, which can be used to remove interferences. The mass filtering quadruple, so this is a quadruple, can filter ions by mass, and then the detector essentially counts how many ions are being detected. And so a lot of these components are really important for ICP-MS analysis. So the sample introduction system consists of a peristaltic pump, which controls the flow of the sample. A constant reproducible flow is crucial for accurate and sensitive analysis. Then the sample is introduced into using a nebulizer. So here, the sample passes through the nebulizer. The liquid sample is converted to a fine spray or aerosol mist, and this is done by a stream of argon that is going into that nebulizer. And so I like to think about this as like a hairspray. And so then once the mist is created, it goes into what's called a spray chamber, which you can see down here. And so anything with large droplets will go to waste, and the rest will enter the plasma in the torch. And so in the plasma is where all the exciting things, I guess, tend to happen. So the plasma that we that is created is with argon gas. So it's an argon plasma, which creates an ionized gas. And here you can see what the plasma kind of looks like. And in this plasma, a lot of things, like I mentioned before, are happening. The small droplets are drying. Then we have decomposition, dissociation, atomization, and then ionization, which is ultimately what the mass spec will be measuring. And the interesting thing about the plasma is that at that temperature, where the plasma is, it is at the same as the temperature of the surface of the sun, so over 10,000 degrees Celsius. And then this degree of ionization for each element, or one in the plasma, depends on the element's ionization energy, or its ionization potential. So how the degree that this sample or this analyte is being ionized depends on the characteristics of the element. And so that also helps or instructs what your limits of detection might be for some of these elements, or one of the determinants for that. So what does it mean to ionize samples? So we have an example of lithium ion, and it's, I guess, sort of a Bohr model, which I guess right now is not really how this is depicted, but what ends up happening is, for the first ionization energy, is how much energy does it take to remove this electron from its outer shell. And so then the element becomes ionized and becomes, in this case, positively charged. So that's one important thing. With respect to ICP-MS and the argon plasma, you are getting cations, mostly cations. And so one of the other reasons why argon is important and is ideal gas for ICP-MS is because its first ionization potential, as we can see here, this is the diagram of the ionization potential, ionization energy for each of the elements, and the percentage of ionization, or how much of that element can be ionized. So you can see argon here has a little bit over 15 electron volts for its first ionization energy. And so most of the elements, all the elements are above, require less energy than that of argon to remove an electron. And then the other nice thing is that this ionization energy is sufficient to ionize, just take one electron, but it's not high enough to take more than one electron. So then we are primarily being able to selectively get positively singly charged species instead of doubly charged species, which will affect some of our measurements. So it simplifies what we're looking at. And you can also see that at 75% of the elements, for the most part, have greater than 75% ionization potential using argon at this temperature, whereas there are a few elements that have not only a higher ionization energy, but also their degree of ionization is lower, such as selenium and sulfur, which then affect the limits of detection. So essentially, you can look at halogens, chlorine, bromine, and iodine. While we can surely measure them by ICP-MS, their limits of detection becomes much more difficult because you have less cations being formed, and it's much harder to remove an electron from these halogens. So then another idea, as I mentioned, most of the elements that we're measuring in this table, you can see is the ionization efficiencies for each element. And so then what this means is how well can you ionize this element. So if you have elements being infused into, and you're measuring, let's say, something like sodium, not only is it highly concentrated in typical food samples and stuff like that, but over 100% of that element is being ionized. So then your limits of detection are not dependent on this ionization efficiency, whereas something like phosphorus, sulfur, or selenium really suffer from the fact that a small percentage of that element is being can be converted into ions. So another issue with ICP-MS in terms of things that you need to consider are interferences. So interferences are essentially, there are two forms that you could find in ICP-MS called isobaric interferences or polyatomic interferences. So isobaric interferences are, for example, you're interested in measuring calcium, and the most abundant isotope is calcium at mass 40. But the issue there is that because we have argon plasma flowing, and this will be at high concentration in our instrument, this argon at 40 also causes interferences with calcium. So it'd be really difficult to measure concentration of calcium when there's so much argon in the environment. Similarly, you can be interested in measuring iron, and the most abundant element isotope for iron is iron 56, but with the a polyatomic species is formed, and so, like I said, the plasma is super energetic, and so a lot of these different species are formed, and so one of them that is formed is this argon oxide that has the same mass as the most abundant ion in iron. So that will also then affect your measurement of iron, and so you would potentially skew your results to declaring that there is more iron that's actually present, because the ICP-MS can't distinguish iron from argon oxide. It could just distinguish the mass that we're looking at. And so I gave you an example of iron and calcium for polyatomic and isobaric, but the periodic table is full of different interferences throughout that you have to be cautious of and be aware of, so that when you are measuring your samples, you're measuring this accurately. And so several elements such as potassium, calcium, iron, selenium, and arsenic have higher detection limits due to plasma-based interferences. But as you recall, selenium and arsenic also tend to have a lower degree of ionization, percentage of element being ionized, and so then that also affects your detection limits as well. So now you have two different things that you have to be aware of with respect to some of the elements and the limits of detection in there. So there are several ways that we can overcome these interferences, and so I'll be talking about a few of these. So we could essentially choose a different isotope of interest. So for example, as prior example, I was talking about calcium. The most abundant isotope is calcium at mass 40, but there's also all these different isotopes of calcium present. And so what you can do is essentially walk and say, well, okay, I know calcium 40 is interfered by with argon, but I could potentially measure calcium isotope at 40 at mass M/Z 44, and then I don't have any interference there. Granted, that is a lower percentage in abundance, so that might affect your limits of detection again, but it is one of the ways that you can overcome this interference. Additionally, calcium in a lot of samples, and you have to know your sample specifically, tends to be a macronutrient in high concentration. So measuring it at an isotope that has a lower relative abundance won't necessarily affect things as badly. Another way that you can correct for interferences or adjust interferences is using correction equations. So naturally occurring isotopes and elements we know of naturally occurring percentages in the earth, and so then we can use predictable formulas to be able to remove some of these interferences. And in these cases, for some elements, this works really well, but for others, there are other things that affect some of these measurements, and so it doesn't work for in all cases. The way that we can further and most routinely get rid of interferences is by what is called the universal cell, the dynamic reaction cell, which helps, which is the feature that we have. And so essentially, we have a small chamber where reactive gas, in our case, we have either oxygen or ammonia. In some cases, in some instances, some people have helium, which is not a reactive gas, but it's a but it is able to remove interferences as well. But in the core, we have reactive gases use oxygen or ammonia. And so in this case, a chamber is basically flowed in with either of the reactive gases. In the case of oxygen, a lot of times what we're doing is we're interested in measuring, for example, selenium-78 has an interference of argon dimer. And adding oxygen gas as a reaction gas results or can result in oxygen reacting with the argon dimer, or additionally, it can react with the selenium. And so now we're measuring selenium oxide, so 78 plus 16 at a different M/Z. And so we have avoided that interference. In other cases with ammonia, what is happening is the reactive gas is reacting with the polyatomic interferences, which tend to be bigger molecules. And so then it allows for the ion of interest, as you can see here, to pass through to the next phase. And so this is a way and a more predictable way of being able to overcome some of these interferences that a lot of times are matrix based in terms of based on what your sample composition is. And so, as I mentioned before, without using the standard, the dynamic reaction cell, you can have something called standard mode. And so typically standard mode, which means no reactive cell is being activated, you can really appreciably know to measure elements of low molecular weight or even high molecular weight. And so because these will not tend to not have any of those interferences that I had indicated before. Whereas most other elements in the periodic table using either ammonia or oxygen are the best way to be able to minimize or remove the interference and accurately measure the element of interest. The next thing that's important to talk about with respect to ICP-MS is the detector. And these are one of the benefits of using ICP-MS for elemental analysis in this case. First of all, I have a depiction here of the different detection limits and the different instruments that are able to analyze elements, and there are positive and negatives for each. But for the most part, I wanted to highlight that ICP-MS covers a 9 orders of magnitude in terms of measurement. So you can measure ultra trace levels of arsenic in the parts per trillion or really low parts per billion level, while also being able to measure the concentration of something like the macronutrient potassium, which is sometimes hundreds of ppms or milligrams in a sample. Additionally, the detector is a dual detector, so it starts off in a pulse for low concentration, so it's measuring really low counts and how many times, and it counts how many times essentially an analyte hits the detector, whereas then as it goes up in concentration, it switches to an analog mode where you can measure higher concentrations, and there are ways that you can calibrate the instrument so you can essentially go from pulse to analog, and so low parts per billion to high parts per million all in the same run if you're interested in a specific element. So we talked a little bit about all the different aspects and components of the instrument, but one of the most important or really primary things that we need to do is due to sample prep. So sample preparation is really important with respect to ICP-MS. There are various different ways that you can do this. There's called open vessel digestion. So I have an example of our crude open vessel digestion that we had when we first started. And so this, you can completely mineralize your samples, but issues are if you have volatile elements such as arsenic or selenium or mercury, this is not, you will be losing some of your element in that way, and so you will not be accurately measuring. The best way to really measure all elements of interest, regardless of whether they are volatile or not, is using a microwave digestion system. And so it's a closed pressure digestion system. Typically what we use is concentrated nitric acid, excuse me, which breaks up the organic material into carbon dioxide, nitrous oxide, and water. And so it's breaking everything down into its elemental form. So I like to think of it as essentially what our digestive system does in our stomach and the acid in our stomach does when breaking down food. Sometimes this is frequently mixed with hydrogen peroxide because it increases the oxidation potential of the reaction and helps break everything down. This releases the elements of the sample so that they could be analyzed by ICP-MS. And then you would dilute the samples to workable concentrations based on the limits of the ICP-MS. And so depending on what elements you want to use, you might have to use a small amount of different acids to help in this digestion. So for example, I have here, if a lot of people are interested in analyzing water, but you need to stabilize water with a little bit of nitric acid, because as you can see here, only the elements in yellow are stable in water to be able to be measured by ICP-MS. So I just want to point out that even though some of these elements are highlighted, doesn't mean that we can measure them with ICP-MS, for example, fluorine, nitrogen, carbon, as an example. And so then if you want to measure water, a lot of times it's good to stabilize, and I tell people to stabilize their samples with a small 1 to 2% amount of volume to volume of nitric acid. Then hydrochloric acid matrices are definitely, hydrochloric acid is really good at solubilizing and these elements dissolving and staying in solution. But remember when we were talking about polyatomic interferences, a lot of these elements form complexes with chlorine, so we want to minimize the amount of chlorine that is in there so that we avoid those interferences. So a lot of times if there are elements that need a little bit of chlorine that need chlorine to stay in solution, we add a little traces of hydrochloric acid to something like nitric acid to be able to solubilize the elements that are needed, but still minimize the potential interferences that occur. And so nitric acid tends to be the acid that we prefer to be used because, as I mentioned before, it tends to reduce any of the polyatomic interferences or interferences that are known to cause issues in ICP-MS. There's another acid that can be used. We don't really use it in our system, is hydrofluoric acid, especially with people who are interested in measuring silicates. You will need hydrofluoric acid, but hydrofluoric acid essentially can melt or dissolve glass. And so a lot of our components in our sample introduction system are made of glass. So there are concerns that need to be addressed. You either have to complex the acid to make sure it's no longer present, or but like I said, we don't really work with hydrofluoric acid. Another acid that is used is sulfuric acid, but that is also just as messy and produces a lot of polyatomic interferences, but just so that you guys are aware of what can be done, sulfuric acid can also be used sometimes with digestion. So this is a summary of all the different essentially periodic tables, and like I said, the gold standard, or what we tend to like to use, is nitric acid, mainly because of the fact that it reduces any of the polyatomic interferences. And if you're interested in specific elements like titanium or stuff like that, sometimes a little bit of hydrochloric acid is enough to keep these in. But a lot of times nitric acid at low concentrations also helps, because a lot of these elements are found in solution in really small quantities. So like I said, sample preparation is super important with respect to ICP-MS. So here we're looking at what type of solutions or solvents can be used for analysis, but we also have to think about purity. As I mentioned before, ICP-MS measures things in ultra trace levels, so parts per trillion, and so we need to make sure that the purity of our sample preparation reagents are also pure. So here's just an example of the purity of nitric acid, ACS grade versus trace metal analysis versus ultra trace. And so for elements that we monitor or are interested in, such as iron, lead, arsenic, you see the high levels of these elements that are present as contaminants in the reagent. And so we want to make sure that we have fairly pure reagents. Excuse me. Ultra trace nitric acid tends to be really expensive, and so we have been able to purchase distilled nitric acid, and we have a nitric acid distillation system. So we twice distill the nitric acid again, and so the purity levels are low enough that will not interfere with our analysis, especially that has not interfered with our analysis. Another thing that's important to consider is the purity of the container. So here is an example from a vendor in terms of the impurities that are found in different types of plastics and tubes that are used primarily for holding your sample. And you can see that low density polyethylene is probably the best type of container to use with respect to impurities, and that increases with the different tubes that you have. But knowing kind of with our experience, we have specific tubes that we tend to recommend for people to use to avoid some of these issues. A few things to keep in mind with respect to sample prep is samples need to have less than 2% dissolved solids. We need it is recommended to prepare sample blanks with all the batches that you're doing. I didn't really talk about this, but use of internal standards is highly recommended. We want final nitric acid concentrations that should be at 2 to 3% volume by volume. We want to minimize the use of hydrochloric acid to avoid interferences. We would like to, with respect to dilutions, use type 1 water with a resistivity of 18.2 megohms, so like millipore type water, to be able to make your dilutions, because that is the purest water in terms of measuring for elemental impurities that it might have. And samples should be within the working concentration of the instrument, preferably microgram per kilogram, parts per billion. But there are ways that when you have a higher concentration of elements that we can adjust certain things to be able to measure those as well. And when in doubt, further dilute your sample. So a lot of times if I'm not sure what the concentration, I would dilute these samples 100-fold and kind of measure it. And so a way to measure these samples is by a technique called total quant. So the total quant method attempts to know everything about the sample from its spectrum. So we are able to, using a small subset of a blank and a standard plus your sample, be able to measure essentially the entire periodic table and kind of give you an estimate of what the concentrations of the elements in that sample are. It is able to using a heuristic algorithm to dynamically choose the best element isotope, depending on the sample being analyzed. It tries to avoid interferences and automatically corrects this, and it calibrates for all elements using information from only a few. And so what this means is, this is typically a method that I use when I have a sample that I want to measure and quantify. And so to give me an idea of what the concentration of the different elements are, I can use this to give me a snapshot of what the concentration of my samples, what the range of my concentration of my samples are for each element. And so how this is done is essentially the instrument samples or scans, masses 6 through 15, 19 through 39, 42 through 210, and 230 to 240. And then again, using a heuristic algorithm, it will give you an output of what the concentration of what the approximate concentration of the elements are. This tends to be actually pretty good estimate, and we can get good if you're not interested in absolute quantification, but you want to have an idea of what the concentration of your sample. This is a technique that could be used to really get you to that idea of the ballpark of where you are. So benefits of total quant: it's a semi-quantitative analysis of about 83 elements, and like I said, it is a good starting point, an option for, like I said, an unknown sample. If people want to measure everything and they're not sure where to start, if you're not sure what you want to measure or what's potentially in the sample, it could give you an idea of what elements are present. And I also tend to use it a lot when I need to identify an approximate range or order of back or where I'm starting point of my calibration curve for each of the different elements. And so, as I mentioned before, the ICP-MS is able to measure a multitude of different elements, starting that are found in high ppm all the way through PPT levels. And so we've been able to develop a method where we are able to analyze 26 elements, including plus internal standards, and in different modes of some of these elements to avoid known interferences that we have developed and that you can use for your samples as well. And so these tend to be elements that we have shown to be of biological importance, both for plant health and human health in this case. And so there are micronutrients such as sodium, magnesium, calcium, potassium, phosphorus, and sulfur. We have micronutrients, iron, copper, zinc. We have some trace elements, as example, barium, cobalt, nickel, lead. And we also have, like I said, internal standards that are in there, which are rhodium, iridium, indium, and the isotope lithium-6. And so I haven't really touched upon about internal standards, but internal standards are very important. Like, for example, in our techniques, we spike our samples with indium from the beginning, from sample prep, and so even with microwave digestion, and then we infuse rhodium, iridium, and lithium-6 so we can correct for variability at different points within the sample process and the analysis. And so this technique has, this method has been published, and I'll talk about some of that research later, but there are some things that we do with respect to limiting variability in ICP-MS. So a lot of times what we do is with our run order, we make a pooled QC. So we add an equal volume of each of our samples. We have a pooled QC, blanks, standards for our standard curve, and we run a pooled QC every 10 injections so that we're able to then perform accurate data processing. And so these different things enable us to do internal standard normalization, blank subtraction, drift correction, and then calculate the concentration of our sample. And so a lot of these different things that we measure help with accuracy. So the difference between a measurement and its true value. And so this we tend to measure by looking at the calibration curve that is run in the beginning and the end, and look at what the potential error is with respect to what the calibration curve is saying is at the end. And then with precision, we're looking at the degree of reproducibility of a set of replicate measurements. So this is expressed as a percent relative standard deviation, percent RSD, or also known as percent coefficient of variance. And so that we tend to measure and be able to measure by our pooled QCs. And so we're able to try to control or correct for a lot of these differences and potential differences throughout our analysis. So here's an example of selenium that was measured. And so this is the run order. And then here you have the counts per second in terms of the raw data. And so the blue dots represent our own unknown samples, and the yellow dots represent our pooled QC. And you see here, without internal standard normalization, the coefficient of variance, or the RSD, is the QC standard deviation divided by the QC average times 100. And you see that RCV is not great. It's 59% in response to the just looking at the raw data. And so then we have a bias factor that we can also calculate, which is the global average minus the QC average divided by QC average times 100. And so our bias here is 33%. So this is not ideal in terms of what we're doing. But if we have our internal standards and are correcting for some of these issues, we can see that as we're correcting with our internal standard, RCV has gone down, and whether we're correcting with rhodium or indium to better levels, and so it has our bias. And so being able to use not only multiple different internal standards to kind of check which might correct for better precision, we're able to, adjust those values in that way, and control and analyze the samples. So here is just an example of essentially the 3 different elements, macronutrients, trace elements, and ultra trace elements, and in terms of how our correction have helped in improving our precision here. And so for calcium, we have now a CV of 3%, copper, same thing, and selenium, we finally ended up with a CV of 9% with all the corrections that we did. And so then you can see how taking into some measurements and some internal standards, we're able to really account for the variability that is present there. That might be, instrument error, or even sample processing error, and we can account for those things and really dig down to what is happening in the samples. So now I wanted to give you 2 examples of how this has been used. So we have looked at potato ionome that has been influenced by market class, and we see that we have 5 different market classes of potatoes: chip, specialty, red, russet, and yellow potatoes. And you could see how the different elements tend to separate and distinguish some of these market classes based on our PCA analysis, which is a principal component analysis, where you basically summarize this data into 2 dimensions, and you're able to explain the percentage of variability in the data based on, PC1 explains 27.47% of the data, whereas PC2 explains 16.93% of the data. And then you could see that if I would to superimpose this on top of this, the scores on top of the loadings, or vice versa, you're able to see which elements tend to correlate more with this separation. So for example, with respect to russet, we see that there's more iron, aluminum, and calcium present in those potatoes. Whereas with the specialty potatoes, we see less zinc, less phosphorus, less potassium. So opposite side is a negative correlation, whereas on the side is positive correlation. We also can look at how each element changes throughout the market class within each market class, and we can see like specific elements that are important for nutrition. For example, we see a lot of calcium in yellow in one of these cultivars of yellow potato. Similarly, a lot of zinc. Our red potatoes seem to have a lot of zinc as well. And like iron, and we have a lot of russets in iron, which we saw in the PCA in terms of correlation. And so that's something else. So using ICP-MS, we were able to see the variation of elements of importance, both toxic and of health, and how they vary throughout. We also did a analysis of elemental profiling of diverse food samples. So essentially 100 different foods that were purchased were done with the Periodic Table of Food Initiative. These samples were digested. We did ICP-MS analysis. We quantified 26 elements, and then with standardized data processing, we're able to see what differences there are. And so some of the highlights are that with the 100 different foods, we're able to see using a hierarchical clustering, we're able to see that, for example, animal products tend to show up here. There are leafy greens, a mixture of fruits and vegetables in both of these, and nuts. But we're able to see different groupings based on the elemental profile that is present. And we're also able to see how these different elements, the macro minerals, trace minerals, other metals, and ultra trace elements, essentially how they distribute throughout the different groups that we were found through the hierarchical clustering, as well as looking at specific elements of importance, such as iron and zinc, calcium, potassium, and phosphorus. And we can even see which foods tend to have more, for example, bok choy seems to have a lot of calcium present by dried food. Beef liver, I guess not surprising, has a lot of iron. So does these prime rib. Cricket protein seems to have a lot of zinc. Egg white from a duck has a lot of selenium. And so these, we can start teasing apart what different components are present in the different foods we eat. And so in conclusion, ICP-MS is available at ARC to analyze elemental composition of your samples. There is a method that has been developed that detects and quantifies 26 micronutrients and trace elements of biological importance in a single run. The instrument can be used across disciplines, and just because this method has been available for 26 elements does not mean that it's only limited to that. Depending on the elements of interest, we can make different methods to be able to analyze those elements of interest to your work. And consideration needs to be taken with respect to sample preparation. Thank you, and any questions?
Thank you, Jackie. We'll open it up for questions.
Hi. Hello, Dr. Chaparro.
Hi.
Wonderful presentation. That was really amazing. And I know you through the seminar, you really spoke a lot about the interferences. So my question was, can there be detection beyond the nominal mass on these instruments, or is the whole for ICP-MS, you just don't go beyond nominal mass?
So right here, so there are some ICP-MSs that are triple quad systems that you can go beyond nominal mass to a certain degree. But we are mass filtering here the quadruple, so nominal mass is really where we are, and so that's why it's been really developed with dynamic reaction chamber and stuff like that as strategies to be able to overcome some of these. And in some cases, some of these methods, like for example, EPA methods, don't actually allow you or let you use like the dynamic reaction chamber and stuff like that for analysis. And so you're limited with respect to some of those methods and to how to report your data. But we're not limited in terms of research that way. And so here, a way to avoid some of these interferences while having nominal mass is using some of these other techniques like the dynamic reaction chamber.
Okay. And the other question I had was, does the ARC facility here have the capabilities of putting an LC before the ICP-MS, just to just try to think of complex organic complexes or something like that?
Yeah. So so for that, for speciation analysis, you would put an LC or a separation technique, whether it's GC or LC, in front of the ICP-MS and use the ICP-MS as a detector to detect after you separate the mixtures. There are some capabilities, but right now that is not present in the ARC. We're trying to figure out how we can potentially bring that for people to use.
Awesome. Thank you so much. Thank you so much.
No problem.
We do have some more time. Other questions? Okay, well, I thank you again, Jackie. That was excellent. And thank you all for coming. And as a reminder to all those in attendance, please do check the website. We will keep it updated and ensure that you register for the next upcoming seminars. Next is Dr. Roy Geis in electron microscopy. And I think we'll hear a little bit about metals during that one as well.
Thank you very much. And if anyone